Hi, thanks everybody for coming. I don't know if Dr. Larch needs a big introduction, but I'm going to give one. He is visiting us from Connecticut University. That's an internal joke for some. I misspelled Connecticut. He received his PhD from Ohio State University in 1994, and he did some postdoctoral work in the University of Pennsylvania. Since the early 90s, he's been working in oscillatory models for originally just for rhythm, and now he has expanded to other domains, I guess, in music development. He has been one of the strongest proponents of oscillator-based models for music perception. He's also editor of several known prestige publications such as Music Perception and Frontiers in Auditory Cognitive Neuroscience. You got that? He has been part of Servas board in different institutions, such as currently he's the president of the SMPC, Society for Music Perception and Cognition. He has received multiple awards, such as the NSF Career Award winner, and also for the Atlantic University Researcher of the Year in 2008, among others. And he's also founder of Circular Logic, music technology company, originally, I guess, in Florida, Boca Raton. And he's going to talk about neurodynamics of music perception, so thanks for coming.
All right, well, thanks. Thanks to everyone who I know, and to Jonathan and George for inviting me. I get to call you George, right? He told me all my friends in Chile call me George, and everyone here calls me Jorge. So I decided to call him.
It always takes bringing you out here for us to suddenly...
So thanks, yeah, and thanks Jonathan, and hello to everyone I'm meeting for the first time. So what I did is I said, well, you know, I'm going to tack this onto this visit to Yosemite to teach at the summer school. So this was a 2-hour talk, and I'm not going to talk for 2 hours, so I may skip a few slides. But I thought there are a number of things that were in that talk. I took a big part of it out, but there are a number of things in there I wanted to share with you. And so this is basically going to be a breadth talk, so I'm going to talk about a number of different sort of applications of nonlinear oscillation to music perception. And so here's my list of graduate students and postdocs who helped me do this work, or sometimes today just do it themselves and then tell me about it later, and my funding agencies. So in 1972, Barlow wrote the following thing. So I'm going to start with my philosophy, a little bit of modeling, just so everyone's on the same page. He says, each single neuron can perform a much more complex and subtle task than had previously been thought. Therefore, maybe the activities of neurons quite simply are thought processes. And this is a perspective which I think from the modern day is a really interesting thing to think about. Of course, David Marr read that and replied in 1982, well, you know, maybe that's not the best way to think of things. Let's, you know, think of this three-level kind of distinction I'll talk about. He said, I want to restrict our attention to the meanings associated with the machines carrying out information processing tasks. Maybe that's the best way to approach it. So we're going to examine a cash register. Okay. And as I'm sure all of you are aware, David Marr proposed this sort of leveled approach to thinking about cognitive processes. So he said, first of all, you develop a computational theory, you decide what's the goal of the cognitive process or the function, maybe from an evolutionary point of view. It's been very popular lately to think that way. So it's the goal, and then what's the representation in the algorithm that we can use to represent that, and then finally, and maybe of less interest to many cognitive scientists, what's the hardware or wetware implementation. And I think this, I guess what I would say is I think this is a very successful way to approach engineering, but maybe not the best way to approach neuroscience. Okay, so that the approach I'm going to take, or I've been taking the past few years, is to sort of go from the bottom up. So say, what do we know about the biophysics of neurons? What can we do using the tools of nonlinear dynamical systems to understand the collective dynamics of maybe a subpopulation of neurons, or the interaction between two populations of neurons, and then further using dynamical analysis, what can we say about the dynamics of behavior, or mostly perception in my case, actually. So rather than, I mean, in one sense, rather than going from top down, we're going from bottom up. But one thing I'll point out is this doesn't really require us to specify what's the goal of the computation, what's the function of the thing we're looking at. We're going to look at it more as a spatiotemporal pattern formation process. Okay, so this is a slide that I borrowed from Eugene Ishikevich, in which he shows a number of different parameter regimes for spiking neurons that are known to exist in real brains. And as you can see, you know, for a single neuron, depending on the ion channels and the way the parameters are set up, you can get all kinds of really distinct behaviors. Now, given this kind of knowledge, you can put this kind of knowledge together with knowledge about connectivity between neurons to make predictions on a larger scale about what populations of neurons do. So many of you might be aware of the Blue Brain project that's currently received, you know, receiving a lot of funding in the European Union. One such project that I found very interesting was a project by Eugene Ishikevich and Jerry Edelman at the Neurosciences Institute, where they tried to do a large-scale simulation of the thalamocortical system. And what they did was they put together fairly realistic models of individual neurons and connected them together into local populations, and connected the local populations together using real human DTI data. Okay, and what they were able to do is produce simulations that are very cool. So if you look over here, and just 1 second, you're going to see a traveling wave arise. Okay. And what they were able to observe in these simulations are traveling waves, oscillations, synchrony of oscillations in different parts of the cortex, and so forth. And it was really just a very impressive piece of work. And I think I would just point out that what the point of that kind of a model is to take our knowledge about the biophysics of neurons and the way these neurons are connected together, and ask how do the local dynamics of local populations emerge from that system, and how did the dynamics of the global dynamics of the brain emerge in interaction with the dynamics of the local populations. Now, as I said, I think that's a really very interesting thing to do, but that, you know, so what it sort of says is, you know, sort of tries to connect the kinds of dynamics we might see in EEG or MEG with the dynamics of neurons. But I'd like to think about the next level up, which is how do the dynamics of perception, cognition, behavior, and affect, and so forth arise from these dynamics? Okay, so really we want to go boom, boom, boom all the way. Okay. Now, I originally put together this slide for a non-musical audience, for a general neuroscience audience, but I've actually since found that it's even more important to show people who are music savvy and to describe what my take on this enterprise is. So why would a neuroscientist want to study music? Okay. Well, for one thing, it's a high-level cognitive capacity. It's a form of acoustic communication. This form of communication is highly structured, so people have compared the complexity of music to the complexity of language. It's universal in the sense that it's found in all human cultures. However, it's also interesting in the sense that music is not the universal language. There's individual universal languages that need to be learned. Okay, so we could study learning and the emergence of language. For the most part, however, you find that music doesn't have referential semantics. So it consists of relationships between self-contained patterns of sound. Now that's actually a really extremely interesting simplification. I mean, and now I'm talking about certain kinds of instrumental music, obviously, but it's a really important kind of a simplification that allows us to consider how the brain is dealing with all these complex structures without worrying about, for example, how it's representing the external world. Okay. Now, and I added this slide added this bullet after the first time I made this slide. This is not to say it's meaningless, right? So music is very important in terms of affective communication, emotion regulation, social coordination, and all kinds of, you know, interesting aspects of social interaction that are still being, you know, uncovered in terms of research. And then I would argue that there are certain universal elements. Any ethnomusicologists in the room? Okay, I don't have to defend myself on that. That doesn't mean that some of these elements that I'll talk about are found in the music of every culture in the entire world, without variation. But there are certain elements, certain proclivities. David Huron was encouraging me just the other day to use the word proclivity instead. That may have their origin in neurodynamics, I think. And so that's what I really want to talk about today. Okay, so what approach am I going to take to music? So first of all, I'm interested in looking at this from a neuroscientific point of view. So let's put up a diagram of a brain, and we've got a cochlea and an auditory nerve and a cochlear nucleus, superior olive, inferior colliculus, lateral lemniscus, auditory thalamus, and auditory cortex, although it's a little bit obscured right on that side. Okay, and now I want to think about this note, though. I mean, a lot of people with a neural point of view will ask what's happening in this part of the brain, what's happening in that part of the brain, and so forth. And of course, that's interesting and important. We have to be aware of that. But I want to think about it in terms of time scale. So what I've done down here is I put a frequency scale, really, that goes from very low frequency. So this is 1/10 of 1 hertz, 1 hertz, 10 hertz, 100 hertz, 1,000 hertz, and so forth. Okay, and I want to think about the time scales of music in relation to the time scales of the brain. So if we looked on the right-hand side of the diagram, obviously you have pitch from about 30 hertz to somewhere about a little bit above 4,000 hertz. We have rhythm beginning at 8 or 10 hertz here and going down. How low? A lot of people think there is a lower limit, but the most careful and most recent investigations by Bruno Ruck have failed to find a reliable lower limit, so I don't want to talk about a lower limit yet. And probably all of you know, but I'll play this simple demonstration that if I take a simple click train and I play it at 200 hertz, that will elicit perception of pitch. Exactly the same click train with a repetition rate 2 orders of magnitude slower gives me the perceptive rhythm, right? And it looks like there's a no man's land in here, sort of. Of course, it doesn't mean we can't hear things in there. So let's play it and see what that sounds like. Okay, so that's 20 hertz. It's a beta band kind of thing. We'll show that beta band in a minute. Maybe a little bit unpleasant. It's kind of the frequency range where if sinusoids interact in that range to produce interference in that range, we might call that roughness. So what about the brain and the auditory system? Well, the basilar membrane creates synchronized responses all the way up to 20,000 hertz. Of course, in the cochlear nucleus, auditory nerve and cochlear nucleus, you will get action potentials, volleys of action potentials, phase locking up over 1,000 hertz. As you ascend and you go to the IC and then the thalamus, what you'll find is that phase locking tends, at least the sinusoids, tends to drop off. Again, we're not quite sure what that lower limit would be according to my physiologist friends. And then if you look in auditory cortex, you find delta band rhythms, which happen to correspond nicely. These 2 hertz rhythms fall nicely into the delta band. You have theta band rhythms, which actually might suggest that there's a second region of rhythm here. Faster subdivisions of the pulse correspond to theta band. Alpha and beta fall into that no man's land, and then you have gamma rhythms in cortex as well. And people, when you get up to these higher frequencies, people have different sort of ways of calling them. Some of them call them high gammas, other kinds of things. But so what I want to argue today is that really the brain resonates to music at multiple time scales, which actually is kind of obvious if you think about it. But I want to stick my neck out a little bit more and say that it's precisely this resonance that leads to musical meaning. So George invited me to talk about rhythm a little bit. So I want to tell you a little bit first about some recent findings in the neuroscience of rhythm, and then talk a little bit about modeling, and then I'm going to go and generalize a little bit. Okay. Okay, so here's a simple musical rhythm. Sounds like this. Okay. And in the hands of Lerdahl and Jackendorf, what they would say is there's a meter. It's got a basic pulse, like that. Okay. There's subdivisions of the pulse, possibly, although maybe not in this particular rhythm, at eighth notes. Then groupings of the pulse into half notes and quarter notes and so forth. Okay. Now, when I started to model these things in terms of oscillations, you know, I would talk to physicists and they say, well, why don't you just do a Fourier analysis? And finally, after years, I said, okay, I'll try it. And it turns out to be pretty interesting, because what you find, for example, is the main beat or pulse at the quarter note level has the most energy when you do a Fourier analysis of the amplitude envelope of that rhythm, or of the onsets in that rhythm. Here, these are onsets actually, so these harmonics of the basic pulse come from the shape of the onsets, because they're kind of sharp. But then you also have energy at these metrical frequencies. Okay. And so it basically more or less agrees with this metrical analysis, maybe with the exception of these harmonics. But then you can ask, what happens if you make this guy a little bit more complex? So let's hear a son clave rhythm. But here the pulse, at least nominally, should be the same 2 Hertz pulse. Okay? But what happens if I look at a Fourier analysis of this rhythm now, is that the amplitude at the pulse frequency has just dropped. Now there are these 2 flanking frequencies that actually have more energy. So why don't people tap here or here? Okay? In fact, actually, sometimes they do. As George and I were remarking earlier. Okay, and excuse me. Okay, so let's make it a little more complicated. Let's go from son clave to rumba clave. Okay, and now it sounds like this. Still should be the same. Okay. And here now we'll find that almost all the energy at the frequency of the pulse is gone. So this is one way of talking about, you know, one of the interesting problems in rhythm is how do you hear a pulse in a rhythm where there's no energy at that frequency, right? So there's something interesting going on there. All right, so 20 years ago now, 20 years ago, almost, I'm a little short of my 20-year graduation date. 20 years ago, I graduated from graduate school. And that's the same year that this paper, Large and Colin, came out, where we suggested that maybe meter could be, you could think of meter in terms of synchronization of oscillations. Then 1999, Mari Jones and I published a dynamic attending theory that modeled attention with nonlinear oscillations. And a few years after that, 2005, or in the early 2000s really, Joel Snyder and I decided to try and do a EEG experiment, and we said, look, if these are really brain oscillations, it should be pretty easy you should see them in EEG. It should be obvious, they should be right there. It shouldn't be hidden, you shouldn't have to infer them, they should just be there. At the same time, we were so here's the way we set up the experiments. We had an isochronous rhythm, so this is literally just the time series, okay? Then we had a rhythm that went loud, soft. And then the key manipulations, at least in our minds at the time, were sometimes we're going to omit one of the loud events, and sometimes we're going to omit the soft events, so there were 4 different conditions. And here's what, you know, sort of Large and Jones or Large and Colin would have predicted. You know, we'd see a single oscillation that synchronizes, and notice the peak of this is anticipating, or sort of, I guess, in this diagram, the onset of that event. Here we might have 2 oscillations that would combine and add, and critically, that we would see events even in the absence of a stimulus. Okay, so that's what we thought we were looking for. this is just the isochronous control condition. Actually, that's interesting enough. I'm not going to show other results because this is an old result. But so what we did, so we were sensitive to the idea that you have evoked responses to sound, and if those evoked responses overlapped, then it would look like there was an oscillation when in fact everything was just an evoked response to the sound. So we decided to look in higher frequency bands. So we looked in frequency band from 20 to 60 hertz, and then there were tones occurring at 0 and 390, okay? And this is part of the montage we recorded, the part that's got the filled circles up there, okay? And we looked at evoked responses and induced responses in EEG. And in FCZ, which is where you normally see a lot of auditory activity, so here it's a representation of the event beginning here and event beginning here, and we saw an evoked response just following about 50 milliseconds after the onset of each event. And in this one even, these are done by subject actually. In this subject we got sort of a strong response followed by a weaker response, which is kind of interesting, maybe subjective rhythmization. But then we saw something here, we seem to see the most power up in this frontal and lateralized electrode, and that tended to be the same over subjects, although not exactly the same place, always more frontal and always lateralized. We saw the peak in power actually preceding by a few milliseconds the onset of the event. And we had a very simple interpretation at the time. We thought that the evoked represented a response to the sound, and the induced response represented the anticipation of the onset. And we saw these evoked responses even in the cases where we left out the stimulus. Okay. Just when that paper came out, I happened to be visiting McGill University, and I sat down with Robert Zatori, and I said, "Where do you think that's coming from?" And he said, "I think it's coming from motor cortex." And I looked at him, I said, "You must be crazy. They're just listening, they're not doing anything." And a couple of years later, his group produced this paper, really Penn-Huen's group, I guess, Chen, Penn-Huen, Zatori, that showed when people simply listen to rhythms, if they have a beat, it activates motor areas of the brain. So I, for one, found that very surprising. I think a lot of people were kind of blown away by that finding. Okay, so a couple years later, Sylvie Nazoridan showed this beautiful result, where she didn't look actually in the higher frequency region, she just looked in the delta band. And did took a steady state approach to analyzing the EEG, and she found with amplitude modulated stimuli, and I'm not going to explain her whole experiment, stimuli were a bit complex, but with just the 2.4 hertz, which is the main frequency of the amplitude modulation, you get a nice response in the delta band at the frequency, the main frequency of the main pulse. And her two other conditions, she said, imagine this grouped into two, and imagine it grouped into three. And really, this is another one of these results that just blew me away. She came to me actually at SFN and said, Isabelle Perret said I should show you this. And I was just like, wow, this is so cool. When they imagine grouped into two, you actually get a frequency in the brain response at the subharmonic. Okay, so what that's telling me now is that it's not only a response to stimulus, but the brain is actually computing with these oscillations, right? And that's how it does this. And then when they imagine three, you get the third subharmonic and then the harmonic of that. So it's just some crazy cool stuff happening. And finally, not everything I'm telling in this story is in the order, but at some point during this array of events, Joel got a postdoc at the Rotman Institute, and he said there are these really interesting people there, so we went up to talk to them. We said, could you do a replication of Large and Snyder for us? And so they designed actually a much better experiment, okay, in which there were multiple frequencies, and there was even a control condition, so it was very cool. And what Takako found is that after the onset of an event, you have this desynchronization in the beta band, and then the resynchronization, the slope of that resynchronization, indexed here by the half max, is really what's anticipating the next event as you change tempo. And that was really very interesting, but the cool thing was that this was done in MEG, and using a seed in auditory cortex, you looked for the other parts of the brain that were also, oh, and I should say this is induced response, and we're looking at the amplitude of beta, of one frequency in the beta band. She looked for other parts of the brain that had this same response and found motor regions. Okay. So now we have a pretty good clue as to what the motor regions are doing. They're also synchronizing at this frequency. Okay. Or more importantly, the beta band processes aren't synchronizing, they're not phase locking, but the amplitude of the beta band is being modulated by this rhythmic input. Then, more recently, I discovered the work of Lakatos and Schroeder in this group. And what they have been doing, and are still doing, I just saw a beautiful talk by Peter. He came to visit us at UConn a few weeks ago. They're recording in the auditory and visual cortices of macaque monkeys. And what this paper is, they've done some other ones, stimulating with auditory stimuli and so forth. But in this particular paper, what they argued is that there's a hierarchy of oscillation in auditory cortex. So that the phase of delta band oscillations, in this case, is modulating the amplitude of theta band oscillations, and then theta modulates higher frequencies and so forth. Okay. Which leads to sort of, I think, a very coherent interpretation, which is consistent with the oscillatory hierarchy in auditory cortex that allows us to see correlates of pulse and meter directly in the delta band, and which are probably, although this hasn't been directly shown yet, modulating these responses in beta and gamma. Okay. And importantly, there's an auditory motor interaction in the beta band. And there are probably lots of other cool things that we don't know yet, but are just around the corner, you know, and you're probably writing them right now. Right? So how do we model this stuff? Well, it's lucky for us that there's a great deal of research coming out of the mathematics and physics communities in terms of understanding nonlinear oscillation in general, of which neural oscillation is just one exemplar. Okay. So one way to tell this story is that neural oscillations, such as cortical oscillations, can arise through the interaction of excitatory and inhibitory subpopulations that are connected sort of in this reciprocal way. And this circuit in general can give rise to lots of different behaviors. For example, an action potential, isolated action potential, or a periodic sequence of action potentials, or rhythmic bursting of action potentials, or even chaotic behavior. Okay. In this talk, we're going to be interested in these two kinds of behavior. Okay. And these happened in different parameter regimes. So we're talking about very specific kinds of behavior in specific kinds of circuits. Okay. The way we model this, so we do build computer models and do theoretical analyses of these models. So we write down equations in which, differential equations in which, one equation is the excitatory population, the other is the inhibitory. And this is a very, very general equation. There are no details in this equation. There are no details about parameters, no details about the dynamics even. Okay, this is just excitation and inhibition. And there's a very general result. Well, actually that's the next slide, sorry. And what we do then is we arrange these into tonatopic frequency gradients. So these are gradient frequency neural networks, or griffins, as we just decided. As an important result of our meeting this afternoon. And so each one of these guys represents an oscillator, and the oscillators are arranged according to frequency, and they might interact amongst themselves. They might project to other griffins and project back and so forth. And this is actually the model. This two-layer model is one I'm going to describe to you about rhythm in a minute. And we stimulate these guys with sound, and we see what happens. Okay. All right. Now, so I started to say there's a lot of amazing work coming out of the math and physics communities over the last 50 or more years. understanding the behavior of neural networks. So this are oscillatory neural networks, or nonlinear oscillators in general. And basically it goes like this. So you take a model of an oscillatory neural network like Wilson-Cowan produced in 1972. And so you've got excitatory and inhibitory action potentials. Sorry, this is population behavior, not action potential. So excitatory and inhibitory populations. You look in state space and you find that it produces sort of, it's a periodic pattern in state space, but it's a funny shaped limit cycle. In this case, it looks like an egg or something. Now, in general, systems like that are very difficult to analyze. They can be analyzed, but you know, it's quite challenging. One very powerful way to look at a system is to do, first of all, a coordinate transformation that takes the funny shaped limit cycle and produces a perfect circle. Okay, so you're really just deforming that and making it into a circle. Once you've made that move, then you've got sines and cosines, you've got trigonometry, you could do the math. Okay, and that's what we do, and that's how we make our predictions. Okay. After the coordinate transformation, what you do is a Taylor expansion of the nonlinearities, and then you just truncate at some point once you've got enough terms to reproduce your basic dynamics. And so this really spectacular result is that this normal form is universal. So anything that is a nonlinear oscillator is described up to a certain level where you truncate by this equation. So this equation describes general properties. Okay. So it's universal. But it's also incomplete. So in the hands of a mathematician, so this is a typical way of writing down the dynamics that you might see in a math paper. By the way, I didn't say, this is still a two-dimensional system because Z is a complex number. Okay, so it's got a real and imaginary part, which you can still think of as excitation and inhibition. So this is the typical equation you might find in a math paper, and they'll write down Z dot, which is derivative of Z with respect to time, equals Z times A plus B, absolute value Z squared, plus some input, and then plus HOT. That stands for higher order terms. That means, we did the Taylor expansion, we just truncated everything over third order. Okay. So we thought about this, and we said, well, actually, if we're going to think about doing general signal processing with systems like this, those higher order terms might be important. So this is something we published in 2010 in Physica D. And what we did is we took those Taylor expansions, we kept all the terms. Okay? And these two things, outlined in cyan here, are sort of summations of those terms. So this summation assumes, so you would normally get here an absolute value Z squared, absolute value Z4, absolute value Z6, all these even order terms. Actually, they're odd order terms because there's a Z out here. So this is actually cubic and quintic and so on. But you just have those all odd order terms in the intrinsic dynamics. That's a universal result. And then there's also this nonlinear function of the input, it turns out, okay, that I've written here, factored into what I call a passive and an active nonlinearity. And I'm going to explain what those do in a minute. Okay. X of t is still the input, and there are a lot of parameters here. The critical one I want to focus your attention on is epsilon. There are a lot of ways to understand what some of these parameters do, but let's just say this. If you hold the amplitude of the input fixed and you vary epsilon, you will increase or decrease the amount of nonlinearity in the system. Okay. So, and the reason I say it that way rather than tell you what happens when I increase or decrease amplitude is just that that's going to be my strategy for fitting data. Is going to be to sort of normalize the amplitude of the input and fit epsilon. Okay. All right. So rather than turn this into a math class because it's only a short lecture, I just want to describe three basic properties of this system. Okay. The first is the property of the intrinsic dynamics. Okay. So if I just turn all the parameters on the nonlinear terms to zero, I have a linear equation. which actually turns out to be sort of a complex generalization of a, complex in the sense of complex numbers, generalization of a gamma tone filter bank, which actually makes it nice to compare with traditional auditory models. And if I plot the strength of the stimulus versus the amplitude of the oscillator, or the absolute value of z, which we call r, then I get a linear function, not surprisingly. Okay. Now I'm going to look at a critical nonlinear oscillator, okay, and we'll tell you what the significance of that is in a minute. I'm going to take the first coefficient of the first nonlinear term, not 0, but alpha, which is the bifurcation parameter, is going to be 0. That's what makes it a critical oscillator. And when it's critical, it means it's poised right between damped oscillation and spontaneous oscillation. Okay? This is the parameter regime that people talk about when they talk about modeling outer hair cells in the cochlea. Okay? So this is, if you will, the amplitude response curve for a cochlear amplifier. So very small amplitudes of stimuli yield relatively large responses, and then very large amplitudes of stimuli get compressed. Okay. Now if I make alpha greater than 0, I will have a spontaneous limit cycle. That means it will oscillate in the absence of input, but beyond that, you have sort of a similar amplitude response. And then by including this quintic term here, I can get another kind of really exotic behavior, which is called a double limit cycle bifurcation, or a Botten bifurcation, or sometimes a subcritical Hopf bifurcation. And what that means is that if I stimulate with a certain amplitude of stimulus, so for low amplitude stimulus, it basically follows this linear curve. But I get to a certain point, and now my amplitude is going to jump up here. And then if I turn the stimulus off, I get a spontaneous limit cycle. So it's a kind of a memory. And I'll exploit that memory for a couple of the models I talk about. So that's intrinsic dynamics. In terms of this nonlinear function of the input, so this passive times active nonlinearity is really sort of a summation of this long series that you see. Now the behavior is as follows. For a small amplitude of the input relative to a function of epsilon, I get a response. So now I've got a frequency gradient. So these are frequency of oscillators from 50 hertz out to 800 hertz. I'm going to stimulate at 200 hertz, and I get a response at 200 hertz. But if I increase the amplitude of the input, now I get responses at harmonics and the second subharmonic. And if I increase even more, I can get even more exotic stuff. So I can get 1 to 1, 2 to 1, 1 to 2, 1 to 3, 3 to 2, all this kind of crazy stuff that people talk about. Again, the advantage of this canonical model is that every one of those peaks corresponds to one term in that expansion. So these are the terms, the x raised to some power, that correspond to the harmonics. So x to the 1 corresponds to 1-to-1 resonance, x to the 2 is 2-to-1, and so forth. The ones with the z bars in them correspond to the subharmonics and to the integer ratios. Okay. Now this simulation, these three curves down there are based on simulations of a critical oscillator. Once that parameter goes above 0 and we have a limit cycle oscillator, something different happens. And I get now, instead of, up to now, these will have the following thing in common with a linear filter. No matter what frequency I stimulate, the oscillator will resonate at that frequency. And it just resonates at greater amplitude when the frequency gets closer. So almost like a linear resonance curve, except that you've got that nonlinear relation. As soon as alpha goes greater than 0, oops, oh, sorry. Okay, so, by the way, notice these terms, okay, that belong to the epsilon, they obey this rule. So where k and m are the integer coefficients of the integer ratio, so the strength of the resonance is epsilon raised to the power k plus m minus 2 over 2. That's going to be extremely important. Okay. All right, sorry about that. So when alpha goes over 0, now we have hard resonance boundaries, or we get these so-called Arnold tongues. So here's 1 to 1, the oscillator is resonating, it's phase locking with a stimulus at the same strength, but now if the relationship between the stimulus and the oscillator is, or the oscillator and the stimulus is 2 to 1, then I get 2 cycles of the oscillator for every 1 cycle of the stimulus. Or I could get 3 to 2. So I get 3 cycles of the oscillator for 2 cycles of the stimulus. Right? Oh, sorry, that's 2 to 1. That's 3 to 2, and then 4 to 3. And those are really the strongest ones, but you can get any integer ratio. Okay, and this is called mode locking. So this is it's a superset of phase locking. So 1 to 1 we refer to as phase locking, but these more exotic ratios are called mode locking. Okay, so this model of neural oscillation is sort of a fundamental property of neurodynamics. So we do see neural oscillations throughout the brain and in different areas, and it could be a single cell, a single neuron oscillating, or it could be these large populations interacting to create oscillations. So we think these are appropriate for studying auditory and music perception. They're based on this canonical or universal model that's one step removed from physiology, which is a slight penalty, but what you get for making that compromise is this analytical and computational tractability. Okay. And what we're doing with these things is we're trying to create a general theory of nonlinear signal processing, or and maybe that's a little too ambitious, a general theory of signal processing with nonlinear oscillators. We're looking at spatial temporal pattern formation in gradient frequency networks, and there's a model of Hebbian plasticity that we're working with. Okay. So how do we use that to model rhythm? I might only get to rhythm today, actually. One of my students, Mark Velasco, set about to study rhythm in the following way. First, he just took a single gradient frequency network of oscillators, where the frequencies here go from 1/4 of 1 hertz to 16 hertz, overshooting the sort of the limit, but that's okay. And he just took one network and stimulated them with a rhythm. And some interesting stuff happened, but the really interesting stuff happened when he took a second network and connected them reciprocally in this way. And these are the relative connectivity patterns. So these are 1 to 1 connections, these are 2 to 1 and 3 to 1. And before we did this model, we had enough experience with the learning algorithm to learn that these are the main connections that are going to develop, regardless of the stimulus rate. Okay, so they were hardwired, but they could be learned, and we're doing some learning simulations now. Okay. Okay, so the following results you get when you have, so you can think of network 1 as an auditory network and network 2 as a motor network. Okay, that's the way we're interpreting this right now. And you get the following results in the motor network. Okay, so here, let me just stimulate it with an isochronous sequence of tones, and this is the Fourier analysis here, so we get a Fourier component at the repetition frequency and its harmonics, and that okay. So the click is the input, and the hi-hat is the response, and you see a nice strong response at 2 hertz. We're only going to play what's happening at 2 hertz. But you also get a harmonic, not surprisingly, because that's part of the input. But now you get a subharmonic. So this would be like spontaneous rhythmization. So it's even in the absence of being instructed, this guy wants to subdivide into 2, or sorry, group into 2. Okay. Okay.
Question?
Yes.
I didn't understand the connections between the 2 networks, between the auditory and the motor.
Okay, so it's a little bit complicated. I did gloss over it, but if it's important to you, that's important to me. Okay, so these are the afferent pattern of connectivity. So the pattern of connectivity going from network 1 to network 2 is just 1 to 1.
Okay, so the output of the oscillator at frequency 1 connects to the input of the oscillator at frequency 1.
Exactly. Okay. The internal connections in both networks are 1 to 1, 2 to 1, 3 to 1, 1 to 2, and 1 to 3.
Okay.
So first and the second and third harmonics and subharmonics, okay? And then the efferent connections coming from the motor network to the auditory network are actually inhibitory. And there's a way to do that.
And those are one-to-one or some other.
Yeah, one-to-one inhibitory.
Thank you.
Okay, rumba clave. Let's try that. There we go. And you want to keep your eye at 2 hertz there. That's the amplitude, and that's the real part, by the way. So it's absolute value of Z, and this is real part of Z. And when it hits like 0.4, it'll start. Okay, so we get to rumba clave, which is a hard one. Okay, that's good. And you see there's only a little bit of energy at 2 hertz in rumba clave, as I showed you before. Now, what we did is we said, wouldn't it be interesting if we could find rhythms that don't have any energy at the pulse frequency? And here's one. They turn out not to be hard to construct. I'll show you how we constructed them in a minute. But here's what the rhythm looks like. And here's a Fourier analysis that shows no energy at 2 hertz. And here's what happens. It's not exotic, just syncopated. So it's pretty clear already that a person would tap at 2 hertz to this. Okay. And this recovers an oscillation at 2 hertz. Okay. And what we find interesting here is that there are also, energy at other frequencies that may predict people's mistakes too. Okay. We're getting 0.5, we're getting a little bit at 1 here, so that's good. So, but, people require convincing, so we wanted to show that people really would tap at the 2 hertz frequency to these rhythms. So we did a behavioral experiment. Our control rhythms were an isochronous rhythm and then rhythms with varying levels of syncopation. So the way we made the missing pulse rhythms is this is an 8-beat framework. And we made 8 events. We put half of them on the beat and half of them off the beat. And then just by phase cancellation, when you do Fourier analysis, you get no energy at the frequency of the pulse. And then these had 3 off the beat, 2 off the beat, 1 off the beat. Okay. And when we asked people to tap, interestingly, they still all tap at 2 hertz, mostly, except some at 1 hertz. So that shows that people really do perceive this 2 hertz pulse. Now, I think that's interesting. I think that's theoretically interesting for a number of reasons. First of all, it completely dissociates the endogenous rhythm with the exogenous rhythm. So if I stimulate a brain at 2 hertz and I see the brain phase lock at 2 hertz, that could easily be a linear filter response that I'm looking at, right? In fact, if I just bang on this table at 2 hertz, it'll resonate at 2 hertz too, right? That's not that surprising. But if I stimulate the brain at a whole bunch of frequencies other than 2 hertz, and it responds at 2 hertz, now I've dissociated those 2 responses. Okay? And I've shown that it's an endogenous cortical oscillation. Well, here, an endogenous behavioral oscillation. We're actually running an MEG experiment at the moment with these 2. Yes?
So, you're not stimulating at 2 hertz in a periodic way. There's phase differences, and that's why there's no 2 hertz signal. But there is, if you look at the spacing between the
That's right.
the two. You have a lot of that. So
That's right.
if there was a
So
phase invariant transformation going on in the processing.
So this won't distinguish between an autocorrelation kind of model and a neural oscillator kind of model. So that's a good point. Okay. So if I would just do an autocorrelation, it would find 2 hertz there.
Right.
So we're running another experiment, or we have run another experiment. I was talking to Kako and George about it, in which we tried to distinguish those 2. And we think we've got some results there, but there are a few issues with that publication at the moment. So, but there are other kinds of stimuli that can distinguish between those 2 as well. Actually, as long as you bring it up, one way to think of a neural oscillation is sort of a neural implementation of an autocorrelation. except it's not merely an implementation of autocorrelation because there are differences, okay, and they can be distinguished. Okay, so I have 10 minutes till we've been going for an hour, and how long would you like me to go?
1 hour 15, I think.
Hour 15, is everybody up for that?
No, that's-
Okay, no one's asleep yet. All right, so one day, many years ago, I was just sitting in my office reading a book about nonlinear oscillation, which is my favorite thing to do, and I said, oh my God, you could actually do general signal processing with nonlinear oscillators. You could develop a theory of signal processing with nonlinear oscillators. And I started to do that. I started to write down the equations and write some code and try and experiment with them. And then I was doing more reading, and I realized that people are already doing this for the cochlea. The cochlea is a bank of nonlinear oscillators. I didn't know that. Its first papers had been just published a couple of years earlier, and I just discovered them. So what I want to tell you about now is my explorations in that area, and we've used these to model cochlea and the subcortical auditory system, or we are engaged in that process. So I want to first tell you a little bit about cochlear dynamics. Our cochlea paper is still in preparation, so I'm not going to show you our results yet, but we're going to use our cochlear model in the next thing, which I will show you. So I'm going to tell you a little bit about cochlear dynamics. Probably everyone has seen this movie. Has anyone not seen this movie? Okay, everyone has seen it. I like to play it anyway because it's so fun to watch. So here's what the cochlea does. I play this, so I'll just tell you a story while you watch it then. So I played this movie in my undergraduate class in auditory perception, and when the questions come about studying the cochlea, they say, well, first you take it out and you unroll it. So it's really all rolled up while it's doing this stuff. And of course, we know that different parts of the cochlea resonate to different frequencies. So any of us who work in these auditory related fields owe incredible debts to these two gentlemen, Helmholtz and von Beckeschi. Helmholtz in 1863 published On Sensations of Tone as a physiological basis for the theory of music, beautiful book, in which he proposed, among other things, a theory of cochlear function, which was a theory until von Beckeschi in the 40s and 50s did groundbreaking experiments measuring real human cadaver cochlear responses to sound. And he, in addition to this, created a very sophisticated partial differential equation model of the cochlea, which earned him the Nobel Prize, and in fact he's the only person who's ever won the Nobel Prize for studying the auditory system, which is pretty amazing, I think. So the combined work of these two gentlemen and many people in between gave us a linear model of the cochlea. So what von Beckeschi found in his experiments was that the cochlea responded linearly to sounds over the physiologically relevant range. So this led to a model that most of us know and love of the cochlea, for example, as a bank of linear filters. So the problem with von Beckeschi's theory is that it turned out to be incorrect. Okay, it's not linear, and already Tommy Gold in the 40s published a paper that said, hey, look, this can't be right. The cochlea can't be linear. For one thing, humans can respond to sounds that are so soft, the energy is the same as thermal noise, right? So if our ear were any more sensitive, we'd hear molecules bumping into one another in an otherwise quiet room. Not only that, the range of hearing is so large that it couldn't be linear. But he made an important prediction. That prediction was this: if he said some biological energy, there's some mechanism of biological energy in there. And he made the following prediction: if this mechanism lost stability, then it would oscillate spontaneously and we'd be able to record the ear-generating sound. Of course, nobody believed that. And Tommy Gold actually went into astrophysics. But in '79, Kemp put a very sensitive microphone into the ears of his subjects and measured spontaneous autoacoustic emissions, which revolutionized our understanding of the cochlea. And now now the dominant model of the cochlea, I would say, is that of in which critical oscillations of hair cells add energy to the vibration of the basilar membrane by growing and shrinking in synchrony with the sound. Okay. Now, as I said, I'm not going to describe our model, all right? But I do want to just show here. These are data measured on living intact cochlea from Mario Ruggiero, showing a couple of signatures of nonlinearity. First of all, the frequency tuning is very tight at low levels of stimulation, very broad at high levels of stimulation, and the place of maximal excitation shifts as the level increases. All key signatures of nonlinear oscillation. And in the late 90s, a couple of key papers appeared. This one, I guess I cited, is in 2000. Manyasko and Hudspeth were very important in the development of this model. And you should recognize this by now as the normal form for the Huff bifurcation. Okay, so this is what I mean by I said, oh, somebody beat me to this. But there's still a lot of work to do in that area. We are working on a paper now with Laurel Carney. And I just want to show you one feature of our model because it's really important for what I'm about to say next. So again, with some data from Mario's lab, here he's stimulating the cochlea with two frequencies F1 and F2, and he's measuring the velocity of one part of the basilar membrane, and what you see is a frequency analysis of that part of the basilar membrane, and it shows response at F1 and F2, but also a bunch of other frequencies, right? And these are well-known distortion products. And there's the results of our model when stimulated with the same stimulus from the Ruggiero lab, and what you see is approximately the same dependence of distortion products on the amplitude of the stimulus. Okay. All right. Now, what's happening in the brain while this sophisticated analysis is happening on the cochlea? Well, one way to answer that question is to look in animals that don't have cochlea. And this is the brain of an African electric fish, Polymeris adspersus. I worked for some time at University of Pennsylvania during my postdoc in Judd Crawford's lab, who studied this fish. And these are the mechanisms of auditory transduction, these gas-filled bladders. Okay, and so they provide some sort of broadband filtering, but not frequency analysis per se. But if you put an electrode into a neuron in the auditory medulla, which is the primary afferent neurons in this animal, and you play a stimulus like this, you can record a response like that. So this neuron itself is frequency sensitive or frequency selective. It doesn't respond to this frequency, but it responds to these two frequencies. Okay. But they're very different frequencies, actually, right? They're almost an octave apart. Here, for one of the frequencies, for the higher of the two that it responds to, you can hear the pitch in the response because it's phase locking, right? It's producing an action potential for every cycle of the stimulus. What about that lower frequency? What's it doing? It's producing two action potentials for every cycle of the stimulus. In other words, it's mode locking. Okay. Now we saw this and reported it. This citation down here is just a poster we did at ARO. It's kind of cool. We had, Devil's Staircase, fit for the for these neurons, but Judd was interested in other things. I didn't quite know what to make of it at the time, so we just let it go. We're actually going to write it. We still got this data. We're going to write a new paper about it. I've got a student working on it right now. But I was particularly interested to see this result from the Sumner lab published in 2011, in which they report mode locking into amplitude modulation of auditory signals in a mammal, in the cochlear nucleus of a mammal. I think it's a gerbil, I actually don't remember. I'm not going to describe this complicated analysis slide, but just to point out that they found subharmonic, a 2 to 3, a 1 to 1, and a harmonic response. Okay. And they found others too, actually. Okay, so briefly to describe a little bit of work in progress, we are working on some models of this kind of response, this mode locking response in cochlear nucleus. We use this so-called class 2 excitable regime, which is basically a spontaneous oscillator. And when you stimulate this with various frequencies of sound, you can get 3 to 1, 2 to 1, 3 to 2, 1 to 1, 3 to 4, 1 to 2, and so forth. And that's the deterministic version, but you can get stochastic versions as well. And now these really start to look like polymerous auditory medulla neurons. Okay. That's just work in progress. What I really want to show you is this model of the auditory brainstem. So in 2009, in Journal of Neuroscience, Lee, Sko, Krause, and Ashley reported brainstem responses using brainstem frequency following responses to musical intervals in musicians and non-musicians. And these are the two musical intervals. They sound like this: consonant, dissonant, according to Lee, Sko, Krause, and Ashley. Don't beat me up about consonant. I don't care. There's two different intervals. Okay. So you see there are electric pianos, so the fundamentals and some harmonics of the fundamentals. The thing that knocked me out of my chair when I saw this was all these different frequencies, in the brainstem response. Where do they come from? Well, sort of the classic answer is they come from distortion products on the cochlea. Right? Well, is that true? How can we figure that out? Maybe modeling can help. Right? So, what we're thinking is, this is the classic model of what's happening in the auditory brainstem. There's phase locking, sure, but that's just sort of a passive response locking to the frequency locked responses in the cochlea, or the phase locked responses in the cochlea. But there are all these complicated circuits in the brainstem, in the cochlear nucleus, lateral lemniscus, inferior colliculus. Maybe those circuits are actually doing something too. Okay. So if it's if it's just this kind of picture, you would expect phase locking. But if it's this kind of picture, you could accommodate mode locking as well. So what we did is we took our canonical model, not that spiking neuron model, our canonical model, and we're trying to model the populations. It's the population models. Okay, so the model looks like this. There's a cochlear layer, a cochlear nucleus layer, and an inferior colliculus / lateral lemniscus layer. And one important thing, the most important thing to tell you about this model and the data is the following. The way this data is recorded is using what's called the polarity reversal technique. So you play the stimulus to the brain, then you reverse the polarity, play the same stimulus, and then you record that hundreds of times, and then you average the two responses together. Now, so you can actually add the reverse polarity, or you can subtract, and people call these the envelope following response and the fine structure response. But from our theoretical point of view, there's something much more precise going on. When you average those two responses, you're removing all the odd order nonlinearities, okay, including the responses to the primaries, which I'll show you in a second. So if it's a primary, if it's a cubic, if it's a quintic, those come out, and you're only left with the even order, giving us a way to sort of subtract out what's happening on the cochlea. So that is shown here. So here are three different layers. The yellow is the single polarity, and the magenta is the average of both polarities, and you see when you average them together, it just completely takes out the response of the cochlea. But the even order nonlinearities restore some of these things, okay? So long story short, I'm leaving myself 10 minutes by your generous allocation of an hour and 15 minutes. I just want to show you that we can indeed predict, and this is a single parameter fitting only epsilon, normalizing the amplitude of stimulus and fitting epsilon. We could predict almost every frequency peak. This frequency peak here that we don't get any energy at all is actually there because it's a difference tone. It's just very weak in our model. So we're still working on little details like that. But overall, we get very strong correlation. So this is R squared of 0.77 and 0.67.
How does that one that doesn't fit map to the frequencies? I mean, what's the relationship?
This is 2F2 minus 3F1. So it's a relatively high order response. And remember that I showed you epsilon to the k plus m minus 2 over 2. If there's more than two frequencies, if there's more than one frequency in the stimulus, there's more than one k. But it's still the summation of those coefficients. So this is a relatively high order thing, and that's why it comes out low in our model. So we still have to think about some of these issues a little bit on this. Okay. I think I'm going to invest 10 minutes in trying to tell you about our model of tonality, because I think that might really interest some people. And it's got some sort of, it utilizes some of the newer discoveries that we've made in nonlinear oscillation. So let me just say what I mean by tonality. Tonality is a way of composing music that utilizes a fixed set of pitches with specific intervals. And when you create a tonal melody, some tones are said to be more stable than others, which generally means they're better placed to end a phrase. And in addition to that, the less stable tones are said to be attracted to the neighboring stable tones. Okay? This general phenomenon, I would say, is cross-culturally universal, meaning it appears in the indigenous music of most cultures, almost all cultures. But tonality isn't the same in every culture, so there's learning involved. Okay? So it's interesting and a little bit challenging to try and think about. There are some examples, but you guys don't need those. Okay, so in the hands of Fred Lerdahl, in his book Tonal Pitch Space, he represents these stability attraction relationships as a hierarchy. So the diatonic tones occupy the second level. These are the non-diatonic tones. These are the tones of the tonic triad, then the tonic and dominant, and finally the tonic is the most stable. Okay, why is this important? So people like Zuckerkandl, I like Zuckerkandl the most. They say things like this. Successions of tones are motions in respect to an order based on forces in tones. What forces in tones? That's crazy, right? It's a very sort of poetic book, written in the 50s. Musical tones point to one another, attract and are attracted. And it's the dynamical quality of musical tones that makes melodies out of successions of tones and music out of acoustical phenomena. This is sort of what makes a melody cohere, a tonal melody cohere. Okay, so it's in another way you can think of it almost as the semantics of tones within a tonal framework. Okay. Now going back 2,500 years or so, the Pythagoreans discovered a kind of relationship between notes in the scale. They summarized it in this symbol, which is called the tetractys, and it symbolizes the relationship between the tonic, the octave, the third, and the fourth in the following way. So the tonic is the 1 or 1-to-1 relationship. The octave is a 2-to-1 relationship. The musical fifth is a 3-to-2, and the fourth is a 4-to-3. And Pythagoras was said to have himself invented the Pythagorean tuning based on the fifth. Okay. And so it's a very sort of ancient relationship. And just intonation was sort of a refinement of that, which is due to Ptolemy. The problem with just intonation is, of course, it doesn't transpose, right? So a major sort of technological advance in Western music was to develop the equal temperament scale, where which sounds almost identical to just intonation, but nothing is an integer ratio. In fact, they're all irrational. Leading Helmholtz to remark, by the way, that the enigma, which about 2,500 years ago Pythagoras proposed to science, why is consonance determined by the ratios of small whole numbers, has been solved by me, Helmholtz. The reason of the rational numerical relations of Pythagoras is to be found in the theorem of Fourier. So basically what he's saying is the is doing Fourier analysis, integer ratios have nothing to do with it, and all this stuff people have been writing for the past 2,500 years about integer ratios is just pseudoscience. Okay? And he goes on to say the relation of whole numbers to consonants became foundation of extravagant and fanciful speculation. Even Kepler couldn't keep himself from this. So he disses Kepler, and then he goes on to diss Euler. Interestingly, this is an important point. He says Euler's system gives no explanation of why a consonant, when slightly out of tune, i.e. equal temperament, sounds just as good as just intonation. Why would that be if it's an integer ratio, right? So this has become, in some ways, and if you read certain literature, you'll find that in some ways this idea of integer ratios has become the very emblem of pseudoscientific hogwash. Okay. But as you see, integer ratios do function into nonlinear, come into nonlinear oscillations. So let's just say, well, wait a minute, okay? And so, I just want to say in that vein that this is not a model of how you should tune a scale. This is a model of what happens in your brain when you listen to frequencies, okay? So I'm not actually going to worry about consonants so much. I'm going to worry about stability. Here's the Krumm-Hansel-Kessler tone profile. Carol Krumm-Hansel, in her book in 1990, asked if Helmholtzian consonants could explain her tone profiles. And she found that while they correlate weakly for the major scale, it's no correlation, no significant correlation for the minor scale. So, but what she did find was that if you look at the statistics of tonal songs, that provides a good correlation. So, and it's been widely investigated since and shown many, many times that the statistics of tonal songs correlate with psychophysical ratings of stability, of tonal stability. Somewhat, I would say, unfortunately, since people have come for moderately good reasons, maybe, but maybe some not so great, to interpret that correlation as a causation, to say that the statistics of tonal song cause the perception of stability. Okay. Well, one way to look at that is to look at cross-cultural perceptions of stability. And in fact, Krumm-Hansel and Barucha and their student Castellano already in 1984 looked at Indian raga. And they actually tested Indian musicians and Western musicians who had no experience with Indian music, which was easier in 1984 than it would be today. But if you just look at the two tonal systems here, I compare the circle of tots with the circle of fifths in Western music, and you see there's actually an interesting relationship. So C major and rag belavel have the same frequencies generally, the same tonic, and then G major and rag or kalyan, this is a tot, not a rag, utilize the same tones. But here, G serves as the tonic, there C serves as the tonic. So it's, comparing to Western music, it would be like a modal kind of a system almost. Okay, so you might expect that people really would perceive stability differently if they had been raised in this culture versus that culture. But when they did the test, oh, sorry, there were some examples. Maybe it's fun to hear those. So, very simple Western melody. Rag belavel, which is same tones, same tonic. Timbre is very different, but maybe you can hear the relationship. Okay, so when they did the study, encultured Indian musicians and unfamiliar Western musicians performed equivalently. There's no significant difference, so they just provided those guys. So we decided maybe phase locking has or mode locking has something to do with it. Now we have two models of this, and this will take me about 2 or 3 more minutes, so we're almost done. I hope you guys are okay. In the first model, we just said, all right, let's provide a very simple model because we can do a closed form solution, and we're going to imagine that the tonic is connected to every other oscillator in the system. So here we have 12 oscillators pictured, okay? So there's just a single connection between the tonic and every other oscillator. And if we write down that system, and we do it, stability analysis of that system, okay, what we find are coupling terms. Now what we're doing is we're picking out the one coupling term that corresponds to that particular integer ratio relationship. That's how we do the analysis. And we find the epsilon coefficient, and again, so what we're going to say is that stability is predicted by this epsilon to the k plus m minus 2 over 2. We're not going to run any simulations. That's sort of complicated, and we'd have to pick all these parameters, and the intrinsic dynamics makes everything behave so differently. Let's just talk about the strength of that resonance that's happening, okay? And let's see if we can predict these relationships that way. Before we worked on the Castellano et al. Paper, I published a book chapter about this in 2010. And it turns out in C major, we can get R squared equals 0.95. Okay? And for C minor, we can get a highly significant correlation that's better than even a lot of the tonal song correlations.
Are all these well tempered?
Yes,
Okay.
yes. Well, no, actually no. Sorry, that's not true. The data is collected with equal tempered scale. Our predictions are based on integer ratios, but I will show you a simulation to back up the validity of this prediction. At the risk of taking a little bit too long, I just want to point out, so that correlation is really great, except that there's a systematic flaw, which is that F, according to this integer ratio idea, should be more stable than E, and it's not. And the same thing happens in the minor scale. Okay? So just hold that thought for a second. Now, but let's use this integer ratio prediction to do the ragas. The yellow is the integer ratio predictions. The magenta are the statistics of the stimuli used in the Castellano et al. Study. And you can see with the naked eye that we're doing a lot better. Okay? And actually, we got Carol and Jamshed Barucha to sign on to this paper, so they are endorsing, or it's still under review, but they're convinced. Okay, and we're doing quite a bit better, in fact, than the statistics. And another analysis showed that statistics actually describes an independent portion of the variance. Okay, but now I really drastically oversimplified to make those predictions and used integer ratio predictions to predict results with an equal tempered scale and so forth. So now let's be more realistic. Let's connect every oscillator to every other oscillator. Let's take into account combination frequencies. Don't even bother to try and read the equations. Okay? And let's turn on our Hebbian synaptic plasticity algorithm. And it's going to learn connections from each oscillator to every other oscillator, and it's also going to learn combination frequencies. So from two oscillators, combination of two oscillators to a third oscillator. Okay. And here's how that's going to work. And now I have 3 movies to play you and I'm done. Okay, so I'm going to train this thing. So now I'm learning the connections too. And I'm going to train it with just a cadence, 4, 5, 7, 1. Okay, and it sounds like this. So that little yellow bar means there's input at that frequency at the moment. Okay, and then it ended on the C major triad, so we got C, E, and G, and then C in the bass. Okay, with some residual activation of frequencies there. And after one training iteration, I've already started to learn some connections. So this is from this oscillator to that oscillator. It's actually, well, it doesn't matter. These are 1-to-1. These are octaves. Okay, nothing significant in this 3-frequency resonance matrix. But now I'm going to just play that over and over again. And I'm going to sort of watch this guy develop. And now I'm learning these are fifths, okay, and there's going to be some fourths and some thirds in that network. This network is kind of interesting because now I've got pairs of oscillators here. I didn't even really know how to label this here. And then going to these oscillators over here. This row happens to be C and G. And notice that C and G activate E. So there are those combination frequencies that go. All right, now I'm gonna skip this one. Okay, and I'm just gonna test this. Now this is the Krumm-Hensel-Kessler profile for the major scale, and I'm gonna just play Twinkle Twinkle Little Star. Never heard it before, it was just trained on the cadences. And watch what, and I'm gonna, these are, by the way, amplitudes of the oscillators, the absolute value of Z that's being displayed there. Okay, and look at what's happening. So this is actually a memory model. I'm using this memory regime, this double limit cycle regime in this network. And you see that basically it's the amplitude of these oscillations in memory that's predicting this tone profile. Okay, and we're getting very good correlations. Here I got 0.94, but the maximum correlation I think went up to 0.99 or something like that at one point after the stimulus went off. And the most important interesting thing that I would point out is that now the relationship between the F and the E are there because of that combination frequency. And then Rog-Balaval is the last thing I want to show you. It's a network trained on the Western cadence. And again, a pretty good correlation that's in memory, as part of memory. So for tonality stuff, I would say that stability and attraction are not metaphors used by music theorists. Possibly they're real dynamic stability and attraction of frequencies in a neural network that's stimulated with tones. And so these musical quality of stability and attraction may be just what it feels like when our brains resonate to music. That would be my contention. Okay, so that maybe statistics don't cause stability, but stability is what causes the statistics to evolve the way they do. So this guy is just based on neural resonance, an analysis of neural resonance plus plasticity, happening at a faster time scale than rhythm. But the whole idea of this thing is that, and maybe I'll put up one more philosophy slide to figure out. So this is neurodynamic approach. It's really different from an engineering approach. Okay, we're trying to make predictions based on the biophysics of neurons, real falsifiable predictions. So it's not as though, okay, I find that my model didn't work out describing this data, I'll just switch out this module and plug something else in. So these are falsifiable predictions that they sort of have to be true by the laws of physics, basically. Otherwise, you're not looking at neural oscillation. Okay, it's not an evolutionary theory. Evolutionary theorizing is so popular now, but am I being recorded? Could you turn off the camera? I mean, it's just to my way of thinking, it's just storytelling. You're telling like a pat story. This is where the behavior is for this, and evolution, somehow sensed that this organism had a problem and designed a solution. I mean, this, and not to say that I don't think that our story needs to be compatible with evolution, because evolution of course happens, but this is based on physics. Okay, and I will point out that physicists like Crick and Prigogine and so forth, when they talked about life, what are the necessary ingredients of life? Evolution, genetics was one half of it, but non-equilibrium thermodynamics is the other half. Pattern formation, entropy. Okay, that's what this is based on. So when my friend David Popple asks, are oscillations the exhaust fumes of cognition? I would say maybe they are cognition. And we just have been looking at it all along, staring us in the face, and maybe with a little help from theory, we can begin to unravel now how they could actually be cognition. Okay, so that's what I have to tell you. Sorry it took so long.
So we have time for a few questions.
How sensitive were your tests to the timbre of the instruments that you use? I can use a pure sine wave instead of something like physical model that has harmonics naturally.
Right. So, not at all, unfortunately. So, this work is being funded, the tonality work is being funded by an NSF grant. And in that grant, we propose we're gonna build the whole thing from the cochlea up, and we're gonna stimulate it with melodies, and we're gonna look at how Indian melodies are different from, how when you learn Indian melodies, you have a different result than Western melodies and so forth. Unfortunately, we haven't gotten there yet. We spent most of our time just unraveling the mathematics underlying this. So for those simulations, what we did is we just decided we'll take what we envision as the highest layer, the memory layer, and we stimulate each oscillator just with a sinusoid at that frequency. So what we're really, and in a way that's not a bad thing to do, because what we really are interested in is what aspects of behavior arise from the interactions of the neurons themselves. And so that's what I'm showing you. Would it be different if there are real tones with real harmonics? Absolutely, absolutely. Hopefully it would be even better, but so that's definitely a simplification that, and you know, we hope to, you know, put all these little pieces that I sort of talked about today together.
I have a question.
Sure.
So in the rhythmic model, you mentioned that the second layer you refer to it as a motor layer. Have you done any experiments like trying to connect it to some inverse model of kinematic model of some part of the body or maybe the head or something? Is there any?
No, unfortunately, we haven't done anything like that yet. You know, one interesting way to experiment with that was proposed to me by Laurel Trainer just the other day at a conference, which is why not do like, and I think she's planning to do this actually, TMS, and so, you know, do some theta burst stimulation, TMS to the motor system, and see what happens to your perception of pulse. And according to this model, so I would say, I actually have a prediction about that, so, which would be, if that frequency is present in the stimulus, you'll see the same, essentially the same thing happening in auditory cortex unchanged. But if you use one of these missing pulse rhythms or just any more syncopated rhythm, you really might disrupt the perception of pulse. So that's what that model would predict, I think. Now, I don't know, I mean, it might be present in the stimulus, the frequency might be present in the stimulus, and you disrupt it anyway when you disrupt motor cortex, which would mean some much deeper interaction is going on.
And I wonder if that's learned, if the motor response might be learned in childhood, dancing, playing music. And if you could find any subjects who've never been exposed to music, whether that would be there.
You know, that's a really interesting question. I mean, I guess the one piece of evidence, the first piece of evidence I would cite would be the Philip Silver studies, you know, where they bounced infants. The only hesitation I have there is I can't remember how old the infants were in that study. But I guess, no, I guess that is good evidence, because they were certainly young enough that they would not be able to synchronize
There's
motor
no
movements.
question
to
they weren't
music.
staying, that's right.
Yeah. So I have no doubt that there are learning components involved in rhythm as well. But I don't think that particular one is one of them, you know, that motor auditory cortex interaction.
Have you tested the same individuals at different times who've been exposed to things to see if there's changes in their response?
Are you, in terms of rhythms?
Yeah, that's this specific.
No, we really just managed to get out this first paper on this missing pulse thing. We did actually, these weren't different individuals or individuals with different amount of exposure, but actually prior to the tapping study, we had used those in an fMRI, combination fMRI EEG study. And we were trying to just, you know, look for responses when there wasn't an event, like originally Snyder and Large did. But we hadn't fully appreciated yet the missing pulse thing. And so we used them in fMRI, and we did sparse sampling. We did after 3 cycles of the rhythm, after 6 cycles of the rhythm, and then we had them rehearse it internally and then reproduce it. And after 3 cycles of the rhythm, interestingly, we didn't get any basal ganglia activation for these missing pulse rhythms. They had to hear it for 6 cycles, and then we got basal ganglia, which was suggestive to me of a time, a relaxation time of this oscillation. It takes a long time for people to hear the pulse here, which is what we see in our simulations. And actually, George was just asking me, is there anything we can do about this? Because it's taken too long to find the pulse. But it seems to me that, naive subjects hearing them for the first time, it takes some time to hear the pulse.
Have you correlated those to meter induction models?
Well, only to our meter induction model. So that's what we're doing now. So we do have a result, maybe I still have it on my computer, or if not, I can bring it up quickly. So, Dropbox, where am I going here? Syncopation, submission. Where is it? There it is. Yeah, it comes up. Do that? Yeah, okay. And let's do this. So what is that? So, is that the one we want? Yeah, that's the one we want. So we had, these are, the simple to complex guys, and we used 5 different stimulus tempis. 2 hertz was the center of the tempo gradient. Wasn't a huge gradient, but here we found 2 significant, 2 effects. Well, actually 1 effect. This one was not significant. So we varied the tempo and we varied the complexity of the rhythm. And according to, by varying tempo, there's no significant effect of relaxation time, which is important for us theoretically for a particular reason. But we also found that complexity, you do get an increase in relaxation time. So this is already 9 seconds. And these are 2 hertz. I think these are, repetition of the rhythm here is 4 seconds. Or, is that right?
Oh, it's 2 seconds.
2 seconds. So this is several repetitions of the rhythm. And actually this is a really difficult experiment because when you rove tempo and you intersperse the different tempi and you get that most impulse thing, it really is hard. And I constructed the rhythms and I was in the experiment and I found it was hard to actually tap the pulse to someone.
Do the people listen to anything before, or is it just silence, or, is there any priming, or?
No, this is just the rhythms presented randomly, going from one to the next.
Because it might be interesting to see if they're listening to something before, how it affects.
That I think that's a very interesting question, and we just haven't gotten to it yet.
Yeah.
So neurodynamics.
Are there any more questions? You want to say something?
I think I've really talked all you guys need to hear.
Well,
Yeah.
thank you so much.